Hello, and welcome to HTAI's annual virtual meeting. We are excited to present What Does Good Look Like? Evaluating Decision Grade RWE Part 2. As we wait for a few minutes for further participants to join, I will share a few slides with you about this webinar. Participants throughout the webinar will remain muted. To submit your comments or questions, please use the questions functions in your GoToWebinar control panel, located on the right-hand side of your screen. Thank you again for joining us. We hope you enjoyed today's session.
Hello, everybody, and welcome back to part 2 of our workshop, What Does Good Look Like? Evaluating Decision Grade Real-World Evidence. A quick recap of who we are. My name is Ashley Jackson. I'm the Vice President of Analytical Solutions at Ation, and my role really focuses on forming partnerships with regulatory and HTA bodies to really help them think through how they're going to use RWE in their decision making. And setting their methodological standards for the use of RWE. I'm joined by Liz Gary, a colleague who is a principal scientist at Ation as well. And prior to joining Ation, she managed an AHRQ-sponsored National Resource Center for Comparative Effectiveness Research Methods. And Liz right now is working both with our clients implementing RWE studies on our platform, as well as with a number of our partners like the FDA. And then we heard from Nicole Gatto yesterday. I don't think she's joining us today, but she is the Senior Vice President of Science. And before joining Ation, she was Epi Group Head at Pfizer. A little housekeeping. Obviously, all three of us are both employed and have an ownership stake in Ation. And we're going to be talking about a little bit more about platforms today and how they can be used for analysis. We specifically did the case study analysis in the Ation evidence platform, but this is really no endorsement of a commercial product. And of course, today we're going to be showing a case study with some results. Please don't share these materials outside of the workshop. Really, the results are, we make no guarantees about the correctness of the findings. We're really using the case study and the results as, a learning opportunity. So they're more for teaching and demonstration purposes only. All right, and so in terms of our agenda, yesterday we had a lecture on describing what makes RWE decision grade, and today we're actually going to hopefully apply those principles and pressure test a RWE case study, a diabetes case study, SGLT2s versus GLP-1s, where we have completed a real-world evidence comparative effectiveness study that we're going to analyze. And then Liz is going to definitely talk through both the study design and the results. And then finally, we're going to do what we're calling a choose your own adventure, where we're going to have a bunch of different options of things that we can test in sensitivity analysis to really pressure test the findings and see if we believe the results and if the results will lead us to making a specific HTA decision or not. So first, we're going to do a poll. We just want to get a sense of who's out there in the audience, really wanting to get a sense of, again, who the members are that have joined the workshop so that we can kind of tailor our discussion and tailor the presentation to the audience. So there might be a little bit of a lag here with the polls, and we're going to be using polls throughout the presentation. This is kind of your ability to, using the polls as your ability to vote on which pathways we take. So there'll be a number of polls, but I'll just pause while you read the options and select the option that is most representative of you, and then we'll move on. All right. So it looks like a number of folks have selected, and it looks like it's a 50-50 breakdown in between folks that are affiliated with a biopharma company and generate evidence for HTA, as well as others. So those others may be academics or students. So that's great. We will kind of take that into account as we're kind of tailoring our discussion. And again, I know that they HTAI said this at the very beginning, but if there's any specific questions throughout the presentation, please type them into us. If there's any pertinent questions on this particular slides that we're on, or any clarifying questions, we will make sure to try to answer those in as real time as possible. All right, let's go back to the slides. And great. Okay, so we're going to go ahead and get started. So the first thing that we want to do is really break down the case study. And the idea here is really to create a hypothetical example of an RWE study and then a subsequent value proposition that might be submitted to an HTA agency for assessment. And the goal is to use this case study to really illustrate the concepts that we learned yesterday during the session on, how do you determine high quality? What is high quality RWE? So we specifically picked a diabetes example. And here in the US, the American Diabetes Association recommends a number of different therapies, a number of different treatment options for second-line therapy, including SGLT2s and GLP-1s. And there's been a decent amount of randomized controlled trials and other real-world evidence studies that have shown that SGLT2s and GLP-1s reduce cardiovascular risk. But this is just kind of one patient-relevant outcome. There are other patient-relevant outcomes as well. And one of those would be treatment intensification. And we're defining treatment intensification as the avoidance of insulin, which is not a preferred therapy among patients. So you could argue that maybe insulin could be cheaper, but there's likely medical costs associated with uncontrolled diabetes. Insulin is usually seen as a last resort treatment, and of course we want to avoid that. And then the route of administration for insulin is less attractive than other drug classes. So the idea of treatment intensification as a patient-relevant or patient-centered outcome is something that we want to study in this particular example. So for today's exercise, we're going to imagine that we're an HTA body and that we've received an RWE study from a manufacturer. And the manufacturer is a manufacturer of an SGLT2. And they want to become the preferred second-line treatment. So what they did is they submitted a comparative effectiveness real-world evidence study that demonstrated a risk reduction in treatment intensification compared to GLP-1s, which is another common second-line therapy. And using what we learned in part one, we're going to pressure test the findings and evaluate the quality of this RWE study. Please note that this is, an oversimplification of the HTA process. One example is that we're looking at a class of drugs here as opposed to one particular product, and that's of course because we didn't want to single out any particular product. So this oversimplification is really, again, just for demonstration purposes only. We just want to make sure that we're learning how to apply the concepts that we discussed yesterday. So the one thing that we have to consider is the level of skepticism that the RWE study and its results will be met with. And this flowchart was developed by Icer in their real-world evidence white paper. It was also adopted by HTAI as well. And the idea here is to understand the evidence and the process standards that, the bar that you need to jump over or you need to meet at the start of an RWE study or its evaluation. So if we can level set and really understand if we adopt the results of the RWE study, if we accept them and we say, yes, SGLT2s should become a preferred second-line therapy, what's going to happen? We're going to expand the use of SGLT2s, and we're going to change clinical practice. So SGLT2s are going to become that preferred second line. So this assertion of superiority over GLP-1s for treatment intensification really requires a high evidence bar, and HTA scrutiny is going to be high. So we really need to ensure that the RWE study is high quality and it is enough to convince an HTA body. So it's a really good thing that we learned about how to generate high quality RWE studies in part 1 so that we can apply it to this particular situation. All right, so I'm going to pass this over to Liz, and she is going to walk through the RWE study design and the results and kind of linking back what we learned in the last section to kind of walk us through, how this quote unquote manufacturer developed this RWE comparative effectiveness study. So Liz, I'm going to pass it to you, but I'll drive the slides.
Great. Hi, everybody. Can you hear me okay?
Yep.
Okay, perfect. Okay. So as Ashley alluded to, this study is SGLT2s versus GLP. And as a reminder, there was a SPACE paper, or a structured pre-approval and post-approval comparative study design framework to generate valid and transparent real-world evidence for regulatory decisions. That's a mouthful. I will only refer to it as SPACE moving forward. But the SPACE paper was presented in yesterday's session, and it really outlines some of the ways to organize and set up your study. And so I'm going to make a lot of parallels back to that SPACE presentation, starting with here. The first step in articulating your design is articulating your research question. And so here, our research question is to compare the effectiveness of initiation of SGLT2 second-line anti-diabetic therapy to GLP between January 2015 and December 2017, and compare on the reduction of treatment intensification. After articulating the research question, really it's important to describe the study design elements in detail. And the study population here is the new users based on a 180-day washout of SGLT2 or GLP-1 that are aged 18 plus, with a diagnosis of type 2 diabetes and no prior use of insulin during the 180 days prior. The treatment here is SGLT2, and the comparator, or active comparator I should specify here, is a GLP-1. The outcome is treatment intensification, which we're defining here as initiation of insulin therapy. And the follow-up starts at treatment initiation and continues until the outcome or the first occurrence of one of the following censoring criteria, which is discontinuation of index therapy, which is defined as a gap in treatment or a gap following exhaust of day supply of one prescription claim, a gap exceeding 60 days until the subsequent claim. Or treatment crossover, an SGLT2 initiating a GLP or a GLP initiating an SGLT2, disenrollment or end of data, which here is September 30th of 2018. Part C rather of articulating the study design is conceptualizing the RCT capture of what we've described in 1B, which is the elements. And so if you want to go to the next slide. So here we want to think about conceptualizing the RCT. And this is either the real RCT that you're trying to emulate, or it can also be a hypothetical target trial that you would theoretically be trying to emulate via your real world design. And so here our real or hypothetical target trial is the enrollment of all diabetes patients who are 18 years or older, who have not achieved glycemic control on metformin alone and have not previously taken SGLT2s or GLP-1s or insulin. and then they get randomly assigned to either SGLT2 or GLP-1. Next slide. So once we have our research question, we've described our design, we've conceptualized the RCT that we're targeting, then we want to start planning our model. And so one of the very first steps of planning a model is conceptualizing the study design. And it's important to do, there's a lot of data-driven techniques to refine your model, but it's important to have an a priori, a model determined a priori before you start really digging into the data. And this is usually based on, extensive literature review, what was done in the trial, what's been done in previous studies, speaking a lot of times with KOLs and clinicians, et cetera. So the most common type of conceptual design for considering your model is a DAG, or a directed acyclic graph. And so here we have a very simplistic version of a DAG showing some of the basic potential confounders that should be considered. And for simplicity, I just in this DAG included more of the categories because obviously once you start putting a ton of different covariates into a DAG, it can become quite complex and very webby. And this is a list of all the covariates that are being considered in this model, including demographics, age, sex, BMI, comorbidities such as hypertension, medication use, ACE, ARB, beta blockers, calcium channel blockers, NSAIDs, and vasodilators, and then various healthcare resource utilization metrics. And again, to Ashley's earlier point, this is by no means an exhaustive list, and most well-designed studies can have upwards of 50 to 100 covariates, depending on what is necessary to control in their model. But again, this is a simplified version for illustration purposes. Next slide. So here's a reminder of the table that was presented that's part of this base paper. Step 2B, it recommends putting together a table where you've actually then take all the variables that you have identified as being important via your DAG and putting them each in column 1 here and then in column 2, there's a description. So here's an example of a table that we've started using as a standard in our research team to kind of take that a little bit further and have kind of a consistent way of documenting the model. And so you'll see in column 1, we have all the variable names. This is, again, not an exhaustive list, but just some examples. In column 2, there's a description. Like taking age, for example, age is typically defined as a continuous value calculated based on actual date of birth. And when you have an RCT or you're conceptualizing an RCT, you would assume that patients at enrollment would have an exact age calculated based on their date of birth. Now, in the market scan data that we're using for this example, in the real world capture, we don't actually have exact date of birth because date of birth is considered for security reasons and patient privacy, we only have year of birth. And so when we calculate age, it's based arbitrarily on a random assignment of July 1st as their date of birth. So we have a pretty close age, but it's not an exact age. And so using the color coding system here, I've marked this variable as an orange, meaning that it can't be exactly replicated in the real world data, but it's pretty close and it's a pretty adequate proxy. And then this last column here we're using as just a checkbox of whether or not we're including the variable in the propensity score model. In the second row, you can see sex. Sex is pretty straightforward, biological sex of male or female. It's reported in the RCT and it's clearly and validly reported in real world data. So it's marked here in green in the color coding, and it is also included in the propensity score model. BMI is a good example of something that, would be included in a likely RCT, but is not something that would be available in the real world data because we typically don't have anthropomorphic type measures like weight and height. And so I've marked it here in red as not available, and it wouldn't be included in the propensity score model. And so I think you guys are probably getting the general idea. This could be a very long list of covariates, but it's nice to also add or consider some sort of color coding as just kind of a visual that can easily be illustrated in conversation with, a regulator or an HTA to make it clear, what are definite greens, what are proxies, and what are things that you just can't capture in the real world data. Next slide. Okay, so this is the study design. I know this was included in a lot of the conversation yesterday. I just want to kind of emphasize here that a picture is really worth 1,000 words, and in some more complex studies, this one picture can actually be representative of multiple pages of a method section of a study documentation. And here for this particular example, we have SGLT2 versus GLP. As a reminder, there's a washout window. The washout window starts at day -180 and it ends on day -1, and it's to confirm whether or not the patient had prior use of SGLT2 or GLP. The exclusions are all applied also during the same 180-day period, although you'll notice that this light blue bar is also overlapping the cohort entry day because we're including day 0 as well in this window. The exclusions here, we're excluding anyone without a prior diagnosis of type 2 diabetes. We're excluding anyone aged less than 18 and anyone with prior insulin use. And then for the covariate assessment period, we're using the same 180-day window. And then as previously described, the follow-up window here in green is censoring patients on discontinuation based on exceeding an allowable gap of 60 days, switching treatment, disenrolling, or the end of the study data. Next slide. Okay, so everything that I've been describing thus far are the details necessary to have study transparency in order to be clear to either a regulatory or an HTA body who's reviewing exactly, not just saying exactly what was done, but why you did each step. And if there's something that wasn't done, why it wasn't done, and really just documenting the journey. And so here's an example of some screenshots that are outputted by the ATM platform, but really just representing the type of or the level of detail, whether it's automatically generated, or whether it's just something that's put together by the author. You can see in the first page, there's a detailed table of contents, breaking down everything that's included in the transcript. On the second page, you can see that you have the methods, describing the methods, starting with a description of the study data that's being used and its fit for purpose. Then on the third page, you can see that there's some description describing, what the exposure groups are. And then lastly, really it's important to know, especially using real world data, exactly how the key components were measured. And this kind of links back to the table we were just talking about. And so not just, oh yes, patients were receiving SGLT2s, it's just really thinking about how you defined SGLT2 using the data source that you utilized. And so here you can see there's a list of generic names that were considered in the measure definition. Next slide. Okay. So based on the study example we did, starting with the primary results, which use a 180-day look back, the 60-day grace period between prescriptions or exhaustion of day supply and the subsequent prescription, we identified a hazard ratio of 0.58, which indicated that patients on SGLT2 were actually at a lower risk of initiating insulin or having treatment intensification than their GLP comparator. When we adjusted for everything in the propensity score model, the estimate got a little bit closer to null at 0.61, keeping in mind that, there's probably some residual confounding here because we used a pretty simplified covariate list, but we can kind of ignore that and brush it away for example purposes. So among the 138,829 initiators of SGLT2 and 98,257 initiators of GLP-1s, there were 96,037 one-to-one propensity score matched pairs that were included in our final results. And this was over a median follow-up of about 216 days, which had an interquartile range of 98 to 429 days. And keep in mind that this is market scan commercial claims. In commercial claims, we often see a lot of churn because patients shop around for the next better deal and might go from insurer to insurer and have shorter longitudinality in the data source. So the value proposition here for an HTA assessment is that the treatment intensification and the avoidance of insulin is a patient relevant outcome. And patients on the SGLT2s had a reduced risk of the treatment intensification, which should lead HTA payers to prioritize SGLT2s as a preferred second line therapy, at least over GLP-1.
Okay. And again, this is that value proposition. This this case study is really an example, a hypothetical example, and is you know, an oversimplification. So we're definitely just using this for demonstration purposes only. Now that we have a real clear understanding, hopefully you have a clear understanding of, you know, what the study design was, how it was executed, whether it's in a platform or not, the transparency behind, you know, what decisions were made and why, and the results and the interpretation of that results. Now that we have kind of that package of, you know, what was done and why and what happened, what were the results, now we can start to think about what types of questions do we have about the data? Where are the uncertainties? If we varied certain parameters, you know, what types of, you know, would it change the result? Would it change the interpretation? So that's what we want to do in this particular part of the presentation. We really want to start to understand, you know, what should we pressure test? And again, for simplification purposes, we kind of created three categories and alternative options of things that we could look at to really pressure test those results to see if they're stable, and to understand, you know, are SGLT2s, should they become the preferred second line therapy? So the three different categories of things that we could vary are across the top here. The primary analysis, which is exactly what we just went over, where our main comparator was GLP-1s. We had the new user washout covariate look-back period of 180 days, and our censoring approach was an as-treated approach with a 60-day grace period. That is kind of our anchoring or referent point. Now we have some alternative assumptions that we can pressure test. And I'm going to go through these. We're going to go through these in a lot more detail, kind of giving you the pros and cons of each option. And then we're going to have you vote on which option you would choose, or what you think we should look at. And then we're going to take that pathway and discuss the results and discuss if that changes the interpretation. So let's get into kind of the options. One of the columns was comparator. And according to our schematic before, the American Diabetes Association listed GLP-1s, SGLT2s, DPP-4s, and TZDs as clinically meaningful comparators. They are the second line choice therapies. We, the study design that we used previously compared SGLT2s to GLP-1s. Now we could argue, one could argue that DPP-4s are another meaningful comparison that we should look at. And you could also argue that it might actually be more meaningful. So DPP-4s are an oral product similar to SGLT2s. GLP-1s are injectables. So DPP-4s may be, you know, in the eyes of an HTA, more appropriate, more meaningful comparator. And this idea of you know, the comparator, not necessarily the comparator that you get, whether it's through a randomized controlled trial or an RWE study, the idea that the comparator might not be the most appropriate comparator, this is something that comes up a lot in HTA assessments. So this is a clear, a natural kind of path of switching the comparator and understanding how SGLT2s match up against DPP-4s. And this is something that's actually really easy to do in RWE, because you don't have to recruit, of course, a whole other treatment arm, and it's of course something that we can do very easily in platforms as well, where it's a few additional measures to create and a few pushes of buttons. But I'll let Liz kind of talk about that once we get to that section. So this is the comparator, switching from GLP-1s to DPP-4s as one particular option that we can pressure test. Another option that we can pressure test and really kind of understand how it may or may not impact the results is how we define who a new user is. So looking at our new user washout and covariate look back period. In a crux, one of the cruxes of defining who a new user is to we have to make sure that we are ruling out patients that had prior use of second-line therapy. And you can do that a number of different ways depending on how you define that look-back period, how big or how small that look-back period is. And as Liz referenced before, dropping or switching insurances, this idea of churn, is really common in the United States. So the look-back period is also very important to powering your study, to making sure that you have enough patients in your cohort to actually get a meaningful result. So in our primary analysis, we used 180 days of look-back period. So what that means is that we looked at the cohort entry date, and then we looked back 180 days to see if those patients were on an SGLT2 or a GLP-1. If they had a fill or a script for one of those drugs, they were excluded from the analysis. So we looked back 180 days for that washout period. Now, we can switch that up. We can switch that up and expand that period to, so not just 180 days, but look back 365 days. And that requires a patient to be followed for a full year before they enter in the cohort. And it's more sensitive to new users because it's going to kick out anybody that's had, a fill for SGLT2 or a GLP-1 during that year time frame. But that's going to also lower the N likely lower the N of the cohort. Another option that we could do, which is even more sensitive, I think, is look at all available look back. So we specify a minimum enrollment criteria. In this particular case, it was 365 days. And then we use all available data for that patient to confirm that they had no prior use of second-line therapy. And this is likely going to yield the smallest patient population or cohort, or N. If we just take a quick example over here on the right, let's take, for example, a patient who had a fill for an SGLT2 or a GLP-1 at day minus 190. So we want to discuss, which one of these three categories, where would the patient be included or excluded? So for our reference or our primary analysis, the patient would be included because that fill is outside of the window, outside of the 180 days. For both the washout, the 365-day washout and the all available look back, that patient would be excluded from the analysis because that fill happened in that look back period that we were interested in. So this is one example of a way that we can kind of alter or change up the study design slightly to understand how robust the results are and if the results hold up across different study design parameters. So this is new user washout covariate look back, but we can go to the third option, or the third category, which would be the censoring approach. And I kind of think of it, the new user look back washout period is you look backwards in time, right? We're looking back to see who used second line therapy. For the censoring approach, I like to think of it as we're looking forward to really understand how long we're going to follow each patient. In the primary analysis, we looked at the as treated approach, meaning we followed everyone until they were either censored or the study period had ended. So that would be this this kind of red or pinkish kind of bar over here. And in the results, Liz did mention a bit about the median follow-up time for the whole study. But if you looked at the median follow-up time by treatment group, SGLT2s or GLP-1s, we saw that there was differential follow-up. So the SGLT2s, the median follow-up for that cohort of patients was 236 days compared to GLP-1 group, which had 188 days. And this could potentially introduce some bias into the results. So one thing that we can test is we can look at an intent to treat approach. And that's actually going to follow patients for unless they're censored, it's going to follow patients for the same amount of time. So this approach is less likely to have differential follow-up than compared to the as treated approach. And so that would be, we would be following patients with the intent to treat 180 days. We would be following patients for 180 days. And so if we take a look at this patient here on the right, let's just assume, they had cohort entry and then they continued to get fills for an SGLT2 past the study end date. In the as treated approach, we would follow them for the time period from cohort entry all the way to study end. But for the intent to treat approach, we would follow them for the study entry or cohort entry date all the way till the max 180 day follow-up time. Alternatively, another option that we could do is to look at if we look at specifically at the as treated approach, we looked at 60-day grace period. And we can extend that grace period to, let's say, 90 days. And this would be a more lenient stance, extending it would be a more lenient stance on the grace period. And again, the grace period is that allowable gap between the end of day supply or the exhaust of day supply till the next subsequent fill. And some patients that stockpile meds or take drug holidays might have large gaps. 60 days might be, some patients might have 60 days, some patients might have 90 days, some patients might have longer gaps. So if we take a more lenient approach on that grace period, we might capture more patients that were on a drug holiday, for example. And this is just another way of varying the study design parameters to really test the robustness of the results. All right, so I think we went through all three of the kind of categories of things that we can pressure test. And of course, there are a lot more. We're selecting these three as for demonstration purposes again. So if we look at all three of those different options, it gives us a number of different combinations of results that we could look at, right? So each row is kind of another iteration, and we see that our main comparator was GLP-1s. In the primary analysis, we looked at 180-day new user washout and a 60-day as treated approach with a 60-day grace period for the censoring approach, and we got hazard ratios depending on if it's matched or unmatched of 0.61 to 0.58 respectively. And so when we vary the different approaches, then we can start to fill in the rest of these columns and rows and understand, how do the results hold up? Does the interpretation change? All right, so I think we're going to launch the first, well, it's the second poll, but it's the first kind of real poll here where we're going to talk about, what do we want to vary? So in order to test the robustness of our findings, what study design choices should we look at first? Should we vary the new user washout covariate lookback period, or should we vary the censoring approach? And again, this is just a poll. There's no right or wrong answer. One thing that we wanted to do is we wanted to start with these two first, because our main comparator is GLP-1s, and so we're testing the robustness of the primary findings with that particular comparator. We will definitely move on to the other comparator, Dpp-4s, later. I think just logistically and logically, it makes sense to test these two options first. So I'm going to let you vote. And it looks like everyone, we want to look at the varying the new user new user washout covariate lookback period. So we are going to jump there. All right, can we go back to the slides, please? Awesome. Okay, so here's just a quick level set. I'll walk you through how this slide is represented. In the top right corner, we see the results of the primary analysis, right? So the main comparator was GLP-1. We had a new user washout of 180 days, and you see our hazard ratios here. Now we have two options for new user washout covariate lookback. We have 365 days or all available lookback. And we have the rationale again as to why we care about this particular options and the pros and cons of the options. So if we kind of go back and remember that churn and dropping in or switching insurances in the US is common. So how we look back is important. How we define that look back period to define new users is going to be important to powering our study and making sure we're sensitive to new users. So we used 180 days in the primary analysis, which was less susceptible to churn and likely to include a larger N, but we could expand that look back to 365 and be more stringent if we did the all available look back. So we're going to launch another poll now to decide which option we should take. Should we look at the option 1, 365 days for the new user washout covariate look back, or should we do all available look back? So we'll give you a few seconds to think about it and vote before we move on. All right. So it looks like overwhelmingly 83% say we should go with the 365 days. So that is what we will do. So we'll go 365 days. All right, this is just the same slide with just our option highlighted here. So Liz, can you talk about how, now that we've decided, okay, we should pressure test the new user washout covariate lookback period, how would we actually accomplish that on in analysis and in a platform?
Right. So yeah, I think it's fine to just leave it all out. So in an automated platform, or if you're using line by line programming and you have macros built, that is also best practice. It's usually as simple as either going to the section on the platform or going to the macro and changing that part. But it's usually just changing that one number from 180 and then in this case to 365. It's important to note that you have to change it in 2 locations here though, because first you want to go back and redefine your cohort in order to apply the washout criteria of what you're defining as a new user. And then subsequently, you want to also then change your covariate assessment period in your analysis plan so that you're also using that same look back period of 365 days. And so unlike some of the other differences that parameter changes we can talk about, this one is a 2-step instead of a 1-step process.
And Liz, question, this just came to me. Is it very common to both have the same new user washout covariate look back period? Is it common to have them both be the same length, or could you argue, or are there situations where you would want one to be longer than the other?
I think generally speaking, the rule of thumb is for consistency, but there might be specific examples where you might want to have a longer look back period for the washout, specifically because maybe it's a drug that's given less frequently. Like if it's an injection or something that is only given, it might be given twice a year. And so, by only having a 6-month look back, it wouldn't be long enough to capture if they got one a little bit longer than 6 months ago, if they're only getting them twice a year. Whereas there might be reasons why you might want to still stick with the shorter look back period for your covariate assessment just because of the restrictions in the data.
Cool. All right, so let's go now to the results. Hopefully this works. Yes, great. So this slide changed slightly. We see that we selected our 365 days, and now we can compare our primary results to the results for this particular option. And you can see that the unmatched results are relatively, unmatched results are relatively similar. We have 0.58 hazard ratio versus 0.57 and 0.61 and 0.62 for the post-matched hazard ratio. And I don't believe this changes our interpretation. Results are relatively stable compared to the 180-day washout. So when we extended the washout to 365 days, the results pretty much remained the same, and our interpretation remained the same as well. So compared to GLP-1s, SGLT2s reduced the risk of treatment intensification by around that 40%. All right, so let's go back now. And
you will notice, just a quick comment there, that it was a pretty negligible difference, but the variance did increase slightly because the sample size decreased when you did the longer look back for 365 days as opposed to 180. So you're excluding more prevalent users that might have made it into the cohort with the 180-day look back for the washout criteria.
Yep. Great. All right. So we looked at number 1. Now let's go ahead and look at varying censoring approach. And again, just to refresh your memory on the 2 different options. So we have, again, the primary analysis up here in the right-hand corner. Our 2 different options were to take an intent to treat approach, so to kind of truncate the max follow-up time to 180 days as opposed to the whole study period. Or we can also look at an as treated approach, but extend the grace period. So instead of a 60-day gap, extend it to a 90-day gap. So a poll just came up, and you'll be able to choose between which option you think we should pressure test. Should we go with the intent to treat? Should we go with the as treated a 90-day grace period instead of 60-day? So we'll give you a second to vote, and then we'll move on. All right, intent to treat it is. Intent to treat 180 days, but it was a little bit closer of a race. So 67% to 33%. All right, let's jump back to the slides. Okay. All right, so let's go. So we decided to do the intent to treat. And Liz, can you talk about how you would implement this type of a change?
Great. So yeah, in this type of a change, we're only having to change one item. And again, if you have an automated platform, this is super, or sorry, actually it is 2 items. If you have an automated platform, this is super straightforward. And here we're showing this screenshot of an analysis plan where we're adding the checkbox for a maximum time reached instead of just allowing the time to keep going until discontinuation or end of data. And we're adding that maximum time of 180 days as a manual entry point. And then we had previously censored on termination of exposure, and we had previously censored on crossover of exposure, and we can go ahead and uncheck those if we want to use the intent to treat design. Similarly, if this was a macro and line-by-line programming, you know, it's definitely a good recommendation to keep things simple so that you can make these types of modifications without having to go back and recode everything.
All right, so let's take a look at our results. So again, up in the top right is our primary results, which we are comparing to the intent to treat 180-day results. So our hazard ratios, again, they've gone closer to the null, slightly closer to the null, where we had an unmatched hazard ratio of 0.58 in our primary analysis, 0.62 in our intent to treat analysis. For the post-match, we had 0.61 as our hazard ratio compared to 0.66. So the results here are still pretty stable. It doesn't really change our interpretation that much. There is a slightly reduction. Well, there's a, closer to the null. So we went from 40% reduction of risk of treatment intensification to around 34 and 38%. Anything to add here, Liz?
No, I think it's really important. Oftentimes when you're doing an as treated approach, when you're following patients to discontinuation or switch, it's important to check if there is differential follow-up between the treatment arms. Because if there is drastic differential follow-up, you may also consider sticking with the as treated design where you're censoring on discontinuation, but you might consider like a max follow-up truncation in order to even things out on follow-up.
Great. All right, so now we can jump back and go to our third option, which is the comparator. So again, here, we talked about this very, a little bit earlier, where we talked about how Dpp-4s you could argue that it is a more clinically meaningful comparator than GLP-1s. One of the reasons is it's an oral product versus GLP-1, which is an injectable. So the idea here is to pressure test, if we had a completely different comparator, would we still favor SGLT2s in terms of treatment intensification? So can you talk, Liz, about how this change is made and how maybe platforms are very useful in such a kind of monumental change, like a crazy study design change?
Definitely. And so again, just taking the example from the platform, the screenshot here, if you have it set up so that you've already have the cohort and you can just copy the cohort and find and replace what was GLP-1 to now Dpp-4, then it actually is a pretty straightforward revision in order to then regenerate the same type and designed cohort, but just with a different comparator choice. because all of your washout criteria, your exclusion criteria, that would all theoretically remain the same.
Yeah, exactly. So our primary analysis for this particular option, our primary analysis stayed the same except for the change to the comparator. So let's go ahead and look at the results here. Again, same setup, top right-hand corner are our primary results. And now we see very different results for the DPP-4s. So instead of with GLP-1s, our hazard ratio for the unmatched and the matched were around 60 or 0.60. Now our for the comparator of DPP-4s, we see a hazard ratio unmatched of 0.97, where the interval crosses the null, and we see the hazard ratio for post-match of 1.03, again with the interval crossing the null. So we get a totally different interpretation here compared to DPP-4s. There's no evidence that SGLT2s reduce the risk of treatment intensification. Liz, anything to add? Any thoughts here?
No, I think it's just important to understand too that we're still talking about the entire cohort of diabetic patients. And in the interest of time, we only chose a few parameters to vary here, but in many circumstances, you would also want to consider various subgroup analyses. And so here, even though we're seeing that SGLT2s are not favored over DPP in the overall cohort, it may be that in certain subsets of the cohort, they'd have a more meaningful impact.
Yeah, definitely a good point. And that's kind of, in the HTA world, HTAs are really interested in those kind of nitty-gritty details about the cohorts because they have the option to recommend with restrictions. So recommend reimbursement for certain smaller subsets of the larger patient population. And so, cohort subgroup analysis is something that could be very useful here. All right. So we saw this before. These were all of the different kind of combinations of the different options. Here in green is our primary analysis. I talked about how we would go about filling this in as we went along. What I'm going to do is I'm going to flash now to just the GLP-1 results so that we can look through all of the different kind of combinations just for GLP-1s, and then we'll move to the DPP-4s because kind of two different research questions, really. How does it compare to GLP-1s? How do they compare to DPP-4s? So for the GLP-1s, again, we varied a few of these options. We only looked in depth at a few of these options, but you can see that the results are again stable, and they were stable in our 2 or 3 examples. But here across the board, you can see that the results hover around the 0.6-ish hazard ratio for the unmatched and the post-matched, with the interpretation that one, the results are relatively robust. No red flags are popping up where something is totally out in left field. And then I think 2, really the interpretation would be that SGLT2s compared to GLP-1s reduce the risk of treatment intensification by around that 40%, give or take, kind of what option you're looking at. Liz, anything to add here?
No, just that again, the point estimate was quite stable in all these scenarios, and this is not a huge variation of a lot of different parameters, but this is the general idea of sensitivity analyses, is that no matter how much you're pressure testing those estimates, that they're not moving substantially in either direction, which would indicate, that there was a potential flaw in the design.
Yep. Yeah. So let's look at now the Dpp-4s, and I think we have kind of a totally different story over here, where the analysis that we looked at in the previous 2 or 3 slides was a hazard ratio of 0.97 or 1.03, depending on if it was unmatched or matched. And then if you look down at varying the number of different parameters that we've talked about today, you could come to different conclusions, right? So if you looked at, for example, all available new user washout and covariate look back with an as treated 60-day grace period censoring approach, you could potentially say that it is protective, that SGLT2s reduce the risk very minimally, but it does reduce the risk. We don't see anything crossing the null here. But if you're looking at the kind of primary analysis just with the Dpp-4s, you're kind of coming to a null conclusion where you can't say one way or the other. So again, I think that kind of highlights the importance of doing sensitivity analysis because you can kind of come to different conclusions. But yeah, Liz, what are your thoughts about the Dpp-4 results?
I mean, I think there's definitely more that can be done to explore, and as I mentioned previously, it's very likely that there's some residual confounding here because we use a very simplified list of covariates in the model. But I think at least it's the results are still indicating that there's some potential decrease in the intensification of the likelihood of intensification to insulin. But at the very least, it is nice to see that we're not seeing any of the opposite effect that would suggest an increase risk and intensification. And so I think I would feel like that helps reinforce the argument, at least that SGLT2s are safe and that they are at least as effective as DPP and definitely not less effective than DPP, at least for this scenario.
Yeah, great. All right. So I think we pretty much have gone over these. Really, the results compared to GLP-1s were relatively stable. And we could say with this particular case study that SGLT2s compared to GLP-1s reduced the risk of treatment intensification by around 32 to 42 or 44% compared to GLP-1s. And that's when we were varying different approaches like the washout period, the censoring, and so forth. And compared to Dpp-4s, it looks like our conclusion could vary from no effect to a slight reduction in risk, depending on what approach we selected. So I think this begs the question to the audience, well, we'll launch another poll. How would you interpret these results in the context of an HTA assessment? Would you or should the HTA body recommend SGLT2s as a preferred second-line therapy over GLP-1s? That was the main study question that we had. So we're going to give you the opportunity to really vote here and see what the audience thinks. All right. So do you consider the RWE study to be high quality given what we learned in part 1 of the lecture? A lot of folks, 40% said yes, 60% could not make a conclusion. There's still some outstanding uncertainties, and I definitely think we highlighted that. You know, Liz, you talked a little bit about some of the uncertainties like unmeasured confounding that we could have because we didn't necessarily use a very robust model. We only looked at a few covariates. But anything surprising here, Liz?
No, I think so. I mean, I think it would be interesting if people could share some of their thoughts about what other what is some of that uncertainty, if it's other than residual confounding, if there are other things. I mean, there's a lot of other things that we could also consider, parameters that could be considered for varying here. And it would be interesting to know if anyone has any additional thoughts there as well.
Yeah. All right. And then our final poll of the day, we'll go ahead and launch that. That is around, do we believe that the RWE study provided sufficient evidence to make a decision? So do we believe that the RWE study allowed us to say, yes, let's change kind of the treatment pathways and let's make SGLT2s the preferred second line therapy? And we'll give you a few seconds here to vote. All right, so this is kind of interesting. Now we say 67% say yes, it gives us a sufficient evidence base to really make a decision, while 33% say no, they cannot make a conclusion, still outstanding uncertainties. So it's a little kind of interesting that the role reversal there from the previous question to this question. So we would, love to have feedback as to if you selected one way or the other. If you have kind of strong feelings about some of the outstanding uncertainties that you think existed, or some additional questions around, why you went one way or the other, I think we would definitely be interested in hearing those. And I think that kind of brings us to the Q&A part. Let's see, I know we did receive some questions from part 1. Liz, do you have any questions on from the audience yet? A few, or because I can go kind of into these questions from part 1 if they're still trickling in.
Why don't we go ahead and start there and see if that inspires some additional questions.
Yeah, so one of the questions that we got from part 1 that that I wasn't able to get to was, can we provide any examples of real-world evidence being used in HTAs? And I think there are a lot of examples. Historically, RWE has been used very, I would say very most commonly for comparative cost effectiveness model inputs. So costs, prevalence, understanding treatment pathways and so forth. But I think recently we're starting to see a shift where real world evidence is being used for more comparative effectiveness, or to discuss the clinical efficacy of a product. And I think there are a few examples very recently where we're seeing a lot more single-arm trials being approved by the FDA and the EMA, we're seeing drugs with single-arm trials being approved by the regulatory bodies, and thus that evidence then trickles down to HTA bodies to try to decide, is this evidence base relevant for reimbursement? we've seen external control arms come out as a potential way to kind of reduce the uncertainty. So one example is Blincyto for pH negative, I think it's ALL. It's a cancer indication where they used a single arm trial and they supplemented, they used an external control arm in real world data to supplement that single arm trial. They provided that both to the regulators and to HTA agencies. And what I found really interesting is that if you look across the HTA agencies, the agencies had slightly different interpretations of the evidence. So some of them were very critical, some of them were very happy with the evidence base. So Blincyto's external control arm for pH negative, I believe it's ALL, is one good example where real world evidence was used to help justify the effectiveness of a product. Another example that I think is interesting as well, and it's very recent, is Icer's sickle cell disease assessment. That particular assessment, I believe it was published in February, March, right before kind of COVID hit the US. And in that sickle cell disease assessment, Icer has committed to actually generating their own RWE. We're actually partnering with them, so that's why I know a bit about this particular example. But what is interesting about that particular case study is that sickle cell disease, there were 3 new products recently launched in the US, and so Icer was reviewing the cost effectiveness of those 3 products. There wasn't really enough data to accumulate for Icer to do, say, a comparative effectiveness analysis. And so one of the challenges of using RWE in an HTA setting is not a whole lot of data has accumulated in order for you to understand the comparative effectiveness of the drugs. But what Icer did is they generated real world evidence to help do a few things. One, test some assumptions that the manufacturer made in their clinical trials. So for example, in the clinical randomized control trials for these particular products, the standard of care, the background use of standard of care was 60-some%, but in the real world data, that standard of care use was a lot lower. It was like 15 or so %. So Icer was able to pressure test, is this a realistic, is the randomized control trial a realistic? Does it reflect kind of the treatment pathways that we're seeing in the real world? And a few other things that they were able to do is use real world data for economic modeling. So they generated specific inputs for the cost effectiveness models. And then they also were able to use the data to really pressure test other assumptions that they had to make in their assessment. So they had a patient survey that they used, and they compared the patient survey demographics. So the patients that participated in the survey, they compared their demographics to the wider RWE population and found it was relatively comparable. So they believe that they had a representative sample. So they were able to feel more confident about using the input from that patient survey into their modeling and so forth. So those are 2 good examples, I think. Icer's sickle cell disease and Blincyto's external control arm. Another question that we got that I think we kind of addressed last time or yesterday is a RWE study, if it's undertaken in the US, is that study kind of applicable to Europe? And Liz, I know that we talked a lot about this idea of, I think you mentioned last time, this idea of fit for purpose data. Do you want to kind of touch on that again?
Yeah, and I think it really does depend on the research question and whether or not there are important cultural or geographical differences in the disease state between the 2. But I think, generally speaking, in most cases, there is a lot of generalizability and applicability between for using US real world data for EMA decisions and vice versa for using European data to help inform US regulatory decisions as well. I think where it becomes kind of trickier is, you know, especially I was mentioning yesterday, when you're starting to go broader and looking like, for example, in Asian countries, there might be either, you know, major genetic differences in the disease area or cultural differences that might restrict the generalizability somewhat. But I think, you know, generally speaking, in most cases, there's a lot of evidence being used from US data for EMA decisions and EU data for regulatory decisions as well.
Yeah, great. And then the last question here is there a checklist that we could create for evaluating RWE studies? So I think that there actually are a number of checklists that have been published. I think Icer published one, and I think there's a few more for evaluating evaluating observational studies or non-randomized studies. But we've kind of started to look through those, and one, there's no gold standard, so there's no one checklist that is recommended by, you know, all reviewers, whether they're regulatory or HTA. And then we've also started to kind of comb through some of these and compare and contrast what elements they're including and not including. And I think there's a large amount of discrepancies or differences between the different types of checklists issued by a number of different groups, whether it's Icer or others. And so I think there's definitely room to create a RWE checklist. And I think that should be done kind of in relationship to the standard setting that the HTAs and the regulatory bodies are currently doing. So they're really defining their methodology and what they're going to recommend in terms of how RWE should be generated and how they're going to be evaluated, how they are going to be evaluating RWE studies. So I think a checklist or some sort of guidance, more comprehensive guidance from those groups is definitely necessary, so that one, manufacturers or researchers kind of know what the end goal is, what the bar is for generating RWE, as well as for HTA folks, regulatory folks, to have a comprehensive understanding of what is expected or what they expect from an RWE study submitted by a manufacturer or researcher. So yes, I think we don't have a checklist. I think there's definitely room, but I think it definitely has to be created in concert with regulatory and HTA bodies as they're starting to set their methodological guidelines and guidance for RWE generation. All right, Liz, I think we're getting close to time. Yeah, any other questions? You're on mute, Liz, I think.
Sorry. There was one question that was a little bit more of a substantive question, and that we talked about the fact, you talked about the fact that it was second line therapy that you were looking at, but I didn't see anything specifying that you were actually making sure that it was second line therapy. Yeah, that's basically the sentiment of it. I don't know if you have any comments there, Ashley, but I think there are two things. First, you know, again, this was a little bit more of a simplistic design. In a perfect world, you might want to also consider restricting as an exclusion criteria, requiring that patients had prior metformin use, showing that they're escalating to second line therapy, as opposed to maybe patients who were receiving the second line therapy as their initial therapy might be somewhat different. Similarly, we might have also considered looking at something like count of prior classes of use as a covariate in the propensity score model. So I think that's a very good point, that we kind of overlooked for the simplistic design, but it is something that might have changed the results slightly.
Yeah, and I think it's interesting that diabetes, it's kind of relatively straightforward, right? Pretty much everyone starts on metformin and then you kind of progress. But for things like cancer, you know, different lines of chemo, and kind of going back to chemo, so I think it can get a lot more complicated. So ensuring that you understand the lines of therapy, and you can define them in the data, I think is a bit challenging for certain therapeutic areas for sure.
Oh yeah, especially in cancer. And I think that really links back to data fit for purpose, and that you have to have the right data source. For example, this example is using MarketScan. MarketScan does not have all the adequate information that you would really need to do a thorough cancer study, such as, staging.
Yeah. Yeah. Great. Well, I think, yeah, I think any other questions, Liz, that came through?
I'm not seeing any others right now.
Okay. Well, there's always the opportunity to email us. I know obviously some folks will be watching this, not watching this live. They will be watching it on the HTAI platform throughout the rest of the year. So if you have any questions, feel free to email us, and hopefully you enjoyed the RWE Choose Your Own Adventure. If you didn't, email us so we know. But if you did, email us as well, because we always like praise. So, Liz, anything else?
No. Thank you all, and thank you, Ashley.
All right. Thanks, everyone. Have a great day.
Thank you.
Bye-bye.